Hello, am I audible to all of you?
Yes, sir.
Okay. I welcome you all in ICHE 2023 at Deshbandhu College, University of Delhi. And let me introduce first today's chairperson of this event, Dr. Sunita Huda. Welcome, ma'am. Uh, Dr. Sunita Huda is working as Associate Professor in Department of Chemistry of Acharya Narendev College, University of Delhi. She did her MSc from University of Delhi in the year 1987, and after that she completed her MTech and PhD from IIT Delhi in the field of chemistry and have published more than 50 papers in international journals. Also, she has completed 5 research projects and has been awarded with awards like Women's Achievement Award, Meritorious Teachers Award by Delhi government, and NISTA Service Award. Uh, Over to you, ma'am.
Thank you. Thank you for nice introduction. Uh, all the best to all the participants for your presentation. Okay. And you must be knowing that we are going to evaluate you on the basis of your presentation. And each one of you will be given, uh, I think there will be 6 minutes for presentation and 1 minute for questioning. Okay. So you have to complete your presentation within a required time period. I think we can start now because the time has already over. We can start with first presentation. Priyanshu.
I request
Hello.
Samarpreet, Sonampreet.
Yes,
Yes,
sir.
you
Here.
can. You can
Please start.
start your presentation.
Okay, okay, ma'am. I hope the screen is visible to all of you.
Yes, screen is visible, and be particular about your time. You
Sure,
are
ma'am.
having this 5 minutes.
So, good afternoon, everyone. Myself, Sonampreet Kaur, belongs to MIT University, Haryana. My topic of the talk is a study yeast breed, Pithecia typhus, and its potential application in bioethanol production. As we all know that this is the era of biofuels, we need to reduce the expenses of normal diesel and petrol, and also our first priority is to reduce the so much of the pollution which caused by such fuels. So here is about the introduction that how Pisces typhus is bioethanol game changer, because this strain has so much of the capabilities and resistance towards so much of the chemical components. The efficient lignocellulosic biomass fermentation capacity is there. The xylose to ethanol conversion with the high yield of the product and the stress tolerance and versatility, a sustainable biofuel solution. So the strain has such of the special genomic characteristics on what basis we can say it is a sturdy yeast. Efficient xylose metabolism possesses genes and pathways for efficient xylose utilization for bioethanol production from lignocellulosic biomass, exhibits the pentose phosphate pathway, which is a robust pathway which maximizes the ethanol yield from the xylose. Stress response genes are there which led the strain to tolerate high level of the stress. Protein secretion machinery, efficient protein secretion for recombinant protein production, and the genetic stability even after so much of the modification. So this is the pathway that how xylose leads to the ethanol conversion. In this kind of pathway, only one step is crucial where I rate isomerase enzyme function. In that step, we need to activate or introduce the starvation pathway, because in some of the condition, the Pithecia typhus yeast cannot utilize xylose as raw. For that, we have to activate starvation pathway so that it can take up the xylose and let the pentose phosphate pathway to lead to the glycolysis pathway to let the ethanol to be secreted in the alcoholic fermentation. So this is overview of our strain with two of them whom we compared as we give 150 gram per liter xylose and it increases yields as from the recorded study it is 30 gram per liter, but we achieved 52 gram per liter in case of NRL Y7124, and in case of CBS 6054 we achieved 45 gram per liter with the 150 gram per liter of the xylose. How we achieve? We introduce two type of mutations. First is the acid hydrolysate mutation. It is type of mutations which prepare the strain to tolerate the acid by keeping it in the different type of temperatures and different type of environment near about from 30 degree to 0.5 degree Celsius. It leads to improve the tolerance of acid, and it has a capability to ferment at very lower pH levels, and the productivity increased about 10 folds, or we can say 1.6 fold to 2.1 fold. This mutant strain demonstrates the performance in acid hydrolysis fermentation, offering potential benefit for bioethanol production. And the second method which we opt is the UVC mutagenesis with the 234 nanometer. Here the thymidine dimers are formed. We can say the covalent bond of thymidine form, which change the purines into the pyrimidines, which leads to the transversion lesions and frame shift mutations to the strain and leads to insertion and deletions of the nucleotides, and these cells are survived about a 40%, and with the survival cells we have to plating it them for the 3 months and 35 degrees Celsius so that we can achieve a novel mutagenesis strain. The another method with the strain CBS 6054 is the SH121 cytochrome 1 gene deletion method. With this method, we have to achieve near about 14 to 16 point mutations. In this modified strain, a specific gene called cytochrome 1 has been intentionally altered or mutated, resulting in the changes to yeast, genetic makeup, or we can say a modification. How it has happened is the cross hybridization can be done. A cytochrome gene, Piscius typhus cytochrome 1, cloned with the wild type modified by cross hybridization with the cytochrome 1 gene from the Saccharomyces cerevisiae. The Piscius typhus Saccharomyces 1 encodes a 333 base pair open reading frame showing 74% and 69% DNA identity to Saccharomyces cerevisiae cytochrome 1 gene and cytochrome 7 respectively. How this happen is we have to utilize the SAM sensitive respiratory pathway. It is an alternative respiratory pathway of salicylic hydrodynamic acid sensitive terminal oxidase pathway, which activates the stress tolerance genes in the particular segment. For that, firstly, we need to prepare the cascade. The cascade is PNQ26, which is prepared by using the EBS to KS vector, and with that type of pathway we lead to the alternative respiratory system which is not coupled with the ATP and leads to production of the cytochrome 1 delta mutant without even altering the cytochrome levels, only deleting the desired cytochrome genes which we need to delete. So how it has happened? And next, it enhances xylose conversion. Mutant produce 21% more ethanol, which is 0.46 gram per gram for 8% xylose compared to the parental strain, which is 0.38 gram per gram. Implications are alternative pathway may be vital for improving xylose to ethanol conversion under oxygen limiting condition. And as we already told you that it uncovered that the 14 point mutations acquired over approximately 7 years of mutations and selection, and we detect that inhibition also in this so that we can ensure that this mutation can be inhibited. So normal petrol and diesel just harm our mother earth. So a very kind of thing. So we need to discontinue it and biofuels. So these are the potential impact, versatility, genetic engineering opportunities, role in integrated biorefineries, contribution to biofuel blending, growing market potential, also fit into a very economical circle. So this is the conclusion of the study yeast Pithecia typhus, that it has effective xylose fermentation, thrives in low oxygen environment, versatile sugar utilization, and also a very evident result, it gives a high ethanol yields, achieve impressively yields near about 84.7% to 90.7%, exceptional efficiency in the ethanol production. So that's all about my topic. So that's why we have to opt the green practices and the green approaches for the production of the biofuel. Thank you.
Okay, thank you. Any question? I want to ask you one thing.
Yes, ma'am.
The work which you have reported, you are saying that it increases the productivity by 2.1 times.
Yes, ma'am.
So, what's the specific reason for increase? Can you suggest?
Ma'am, actually, from the reported studies or the previous study, it was only 1.4% of the increase. And
Mm-hmm.
when very first we detect, it is 2.1. And in
Okay.
our ongoing work, which I can't, due to some of the
So you are
copyright issue,
saying
I
very
can't
specific
disclose, it
2.1.
shows
That's why
us the
I'm
near about.
asking. Yeah. Uh-huh.
Yes, ma'am. Yes, ma'am. That is already reported in our paper, and which is not reported, that is, it is increased up to 420 to 40%. And now we are on the pathway to develop a pure bioethanol engine with
Uh,
the yield of high yield. Yes, ma'am.
okay. Thank you. Thank you. All the best.
Thank you, ma'am.
Uh, Now the next candidate is Anand Kumar Yadav. Is he here?
Yes, ma'am.
Okay. Start.
Once minute.
Be particular about time. You're having just 5 minutes.
Ma'am, can you call my computer hang? Can you call next candidate? I will give after that.
Okay. Next is Girja.
Okay, ma'am.
Okay. Start, please. Girja, try to finish within 5 minutes.
Okay. I, Girija PV, Department of Chemistry, Calicut University. My topic is UPLC, ESI, QTOF, MS, Profiling of Prasannadi Kashaya.
Beta, your slides are not visible.
Slides are not visible. Why? I share it now.
I don't know. I'm not able to see it.
Why? I don't know. What's going on?
You have shared your screen as giving OB53. You have to click it, I think. Your presentation is, your number is 53 now.
Yes.
Then you have to click it. Your presentation is not there on the
Now
screen.
it is the same?
No.
No. Why?
Are you able to see rest of the candidate?
Yes, I can.
No, I'm not able to see you.
Why?
Your slides are not visible, Girja.
Okay. That is screen share.
No, only Priyanshu Prasad is available. He's visible.
Dr. Anil this side, ma'am, from Priyanshu's PC.
Okay. Your screen is with different name.
Yes. Sir.
Yeah.
Now is it seen?
No, beta, you are not
Yes,
visible.
I don't know what is going on.
Can we start with the next candidate? Because we are running short of time.
Ma'am,
Okay. Okay.
Anand,
Okay, ma'am.
Mr. Anand is there, I think.
Yeah.
Anand, you can start.
yeah,
Anand,
ma'am.
you can start, beta.
Okay, ma'am. My work is related to the removal of dye using biochar.
Now,
So
beta, share to karo.
yeah,
Share
ma'am.
your screen.
I shared. Once minute, ma'am.
Girija,
Anil, are you able to see?
it is still Girija's screen, I think.
Again, the screen is
Yeah.
coming. I'm not able to see. What happened?
It's on. I shared. Yeah, in my laptop, it's saying you are screen sharing and the topic is on like presentation is open.
No, Anand, I'm not able to see you. Your PPT are not visible. What about Anil? Are you able to see PPT?
No, no, ma'am, no.
Yeah.
It is still Girija's screen.
Your
Anand
screen
Kumar
share
has
is
started
reloading.
sharing. No, the message appeared, but I'm not able to see.
Ma'am, it's saying you are screen sharing. I think it is loading.
Girja has to stop first. Someone has written. Girja, beta, you have to stop sharing. Then only it is visible. I
Yes,
think your
I
screen.
stopped now.
Girja's screen is visible only. Yes. Now Anand Kumar's screen is visible.
Okay, ma'am.
Thank you
Okay.
so much. Someone has pointed out this thing and then the matter is resolved.
Mm.
Yes, you can start.
So, ma'am, my topic is the development of corn store biochar for removal of dye from water. So the thing is ki, today's we are like facing water shortage, and it is said that by 2050, uh, a lot of people, around 1.7 to 2.4 billion, will face water shortage. So my so the water pollution caused by the presence of a lot of chemical waste, plant nutrients, fertilizers, so they need to be addressed properly because they are degrading the water quality. And dye among them is one of the most toxic, carcinogenic, and mutagenic organic pollutant. Every year, more than one, more than 7,000, uh, more than 3 lakhs tons of dye are produced, especially in India, especially in for a financial year of 2022. And if we dye 1 kg of cotton, it usually requires 70 to 150 liters of water and around 30 to 40 grams of dye stuff. So on an estimate, it is said that 280,000 tons of dye they discharge in water. They are usually are inefficient due to inefficient textile dyeing and all. So on certain 10 to 200 ppm of concentration has been observed after after textile industry discharge the water in if discharge the water as effluent. And these are the few major cities where India has in India that are textile hub, and there is the problem of dyed wastewater. And the risk associated with this dyes wastewater is like for human, it's like carcinogenic, mutagenic, and chemical teratogenic, skin irritation, bioaccumulation. These are the few risks associated to the humans and animal. And aquatic for aquatic species, they usually uh, uh, they usually interrupt the sunlight, and that's why photosynthesis process is like inhibited. And these are like few typical characteristics of textile effluents. And the color standard set by CPCB, it's like 150 platinum platinum color units, platinum cobalt color units. And these are the distribution of synthetic dye in aquatic system. Okay, how these dyes are like can degrade in leucoform, are aromatic amines, then sedimentate through sedimentation and immobilization can develop on the surface, and then it through bioaccumulation, fish and other species can eat it, and then it biomagnifies in humans and animals. So these are the few methods that are widely popular for textile effluent treatment plants. But I choose only this physico-chemical method because these are like very simple. Uh, these are chosen due to their simplicity, their low cost, their excellent removal, and no secondary contamination. So I chose these two dyes to remove, like crystal violet. It is usually it biodegradation product are way harmful, like aromatic compounds and amines, and these are like few human uh, few risk associated to the human. And there is acid orange 7, which is like anionic dye, and this crystal violet was like cationic dye. So I choose two categories, uh, and what I did is like I dried some corn store biomass, then chopped it well, and then passed through its muffle furnace. Then for retention time of 1 hour at 700 degrees centigrade without oxygen, and these are the like pyrolyzed material. Then I through particle size I choose different particle size. However, at the particle size mentioned here, 52 to 100 bss, the adsorption found to be good. So these are the few characterization of my biochar, where I found out the yield is like 24% and S content was like 6.62%. And these are the like carbon, hydrogen, nitrogen, sulfur, and oxygen ratios in percentage. And the second is the PZC 0.0 charge. Below this point, the biochar is present with a very positive surface area, and after this point, it presents like negative surface area. Then I characterizes their presence of few elements at the surface of the biochar through same EDS. And like we can found carbon, oxygen, magnesium, and silicon and calcium are there on the surface of the biochar. Then these are the few surface morphology of the biochar that I developed, where we can see there are some macroporous morphology where with grooves and voids. This indicates the broad range porosity, and that's why it is suitable for rapid dye sorption. And second slide. Then there are few batch studies I did, dose kinetics as well as concentration kinetics. You can see these are the graphs, and the R square value was found to be maximum for pseudo second order reaction. So there is involvement of both concentration of dye as well as the dose of the biochar for reaction mechanism, dye uptake reactions. And next slide is like concentration kinetics. Again, in this kinetics also it is
Try to
found
wind
that
up, Beta. Time is over almost.
okay, ma'am. So in isotherm study, I do Langmuir and Fendlick isotherm studies, and I found that at 25 degrees, it's like 64 milligram per gram. And for RN7 dye, since it is negatively charged, and the adsorption capacity found to be low, around 6. And these are the column study that I performed, and these are the parameters such as exhaustion time, flow rate, total mass जो pass हुआ, total adsorbent mass, and percentage removal. And capacity, column capacity usually comes out to be lower than the batch study because a lot of the biochar particle are not come in proper contact with they don't have like proper contact time as we do in batch. So it's like 25 milligram per gram for crystal violet and 1.83 gram for milligram per gram for acid orange 7. And these are the few adsorbent that are available that has been researched, and I found my work to be incomparable to those adsorbent. So you can see. So final conclusion is that it is a sustainable and eco-friendly solution and easily prepared, and they have high capacity as well as the cost is very cheap. So it can revolutionize the wastewater treatment plant field. Thank you, ma'am.
Which dye is getting more absorbed on your biochar?
It's crystal violet dye, ma'am. Usually
Why?
biochar surface is negatively charged.
Okay.
So that's why.
Any other question? Anyone want to ask any question? Okay, thank you, Anand. You can leave the screen now. Stop
Okay.
sharing the screen, then only other can start.
Okay.
Girja?
Okay, ma'am.
Yeah,
I
you
will
can
start
start.
now. I Gidja PV. My topic is UPLC ESI Cute of MS Profiling of Prasannadi Kashayam. Prasannadi Kashayam is one of the Ayurvedic
Again, your
polyherbal.
slides are not visible.
Now also?
No.
Ma'am, maybe she's sharing different screen. There, that slide may be open, but it's like on different screen. There is option to choose.
Okay.
Oh, other screen is not visible.
Yeah.
Oh,
Now
yeah.
yeah.
Yes.
Okay. My topic is UPLC ESI Cute of MS Profiling of Prasannadi Kashayam. Prasannadi Kashayam is one of the Ayurvedic polyherbal formulation used as anti-inflammatory drugs. It is mainly prescribed for shoulder-related disease. As per Sahasrayogam, it is the decoction of 6 plants. They are Allium, Sativum, Alpinia calcarata, Meremia tridentata, Sida rombifolia, Vigna mungo. What is anti-inflammatory drug? Anti-inflammatory drug is a drug which reduces inflammation. What is inflammation? Inflammation is a defense mechanism in the body. It is part of complex biological response of body tissues to harmful stimuli such as pathogen, damaged cells, or irritants. The function of inflammation is to eliminate the initial cause of cell injury, clear out necrotic cells and tissues damaged from the original insult, and initiate tissue repair. Inflammation are of 2 types, acute inflammation and chronic inflammation. Acute inflammation is a short-term response with a localized defect as it works on at the place where a problem existed. Chronic inflammation can have long-term and whole body effect, and it is the reason for the chronic disease such as depression, cancer, heart disease, diabetes, psoriasis, allergy, etc. So inflammation needs to be well managed. Anti-inflammatory drugs nowadays, modern medicine use steroids and non-steroidal anti-inflammatory drugs. Patients prescribed with steroids and non-steroidal anti-inflammatory drugs experience mainly gastrointestinal and renal side effects such as dyspepsia, gastric ulceration, high blood pressure, osteoporosis. So because of this, all they opt Ayurvedic medicine in which side effects are absent or minimal. Polyherbal formulation. Ayurvedic medicines are mainly based on plants. About 90% Ayurvedic medicines are polyherbal formulations. Polyherbal formulations are the formulations prepared from more than 3 plant parts. When synergism is considered, polyherbal formulation offer when synergism is considered, polyherbal formulation is very effective compared to single herb formulations. For this, lower dose of herbal preparation may be needed to cure the disease without any side effects. These benefits have resulted in the popularity of polyherbal formulation. Importance of Ayurvedic medicine. Most chronic diseases are multigenic and hence multi-targeted approaches required to ameliorate or cure the condition. Unlike the allopathic system, which uses mainly synthetic chemicals designed for specific target receptors that primarily give symptomatic relief, Ayurveda involves the use of natural products to eliminate the root cause of disease by restoring balance. Why this study? The activity of Ayurvedic formulation is due to the presence of phytochemicals in them. Ayurvedic formulations are in Ayurvedic formulations, pharmacological principle works together in a dynamic way and produce maximum therapeutic efficacy with minimum side effects. Ayurvedic medicines exist in different formats, including decoction, powder, fermented products, tablet, and medicinal butters. The kashayam are the decoction of plant part. During the preparation of kashayam, different reactions may occur. So the kashayam is a complex mixture of phytochemicals. Identification of compounds from such a complex mixture is possible by LCMS only. The present study is the identification of compounds present in Prasannadi Kashayam UP Ultra Performance Liquid Chromatography ESI Q-TOF MS. LCMS is a hyphenated technique comprised of chromatography and mass spectrometry. LCMS provides identification of unknown compounds through efficient separation capabilities of HPLC and exact structural information by mass spectrometer. UPLC coupled with ESIMS have provided excellent method for identification and isolation of a new secondary metabolites from complex extracts. Experimental part 300 ml of well-shaken kashayam is centrifuged at 4000 rpm in a REMI HC centrifuge for 30 minutes. The clear supernatant was carefully decanted and this was analyzed for UPLC Q-TOF MS. The experimental conditions are given. CAT column is used. Then mobile phase used is 0.1% formic acid in water and acetonitrile. The result from the elemental analysis and mass fragmentation, 19 compounds have been identified. Out of 19 compounds, 4 are terpenes, 5 alkaloids, 10 phenolic compounds. They are given in the table all with its mass fragments. The reported literature work, 97% identified compounds are used as anti-inflammatory or have a property as anti-inflammatory and antioxidant. So, conclusion, UPLC Q-TOF analysis conducted on the supernatant of the kashayam showed the presence of 10 phenolics, 5 alkaloids, and 4 terpenes. These alkaloids and phenolics and terpenes are most characterized phytochemicals for antioxidant and anti-inflammatory activity. So the kashayam possessing these phytochemicals have served as a source of potent antioxidant and anti-inflammatory agents. This study has validated anti-inflammatory property of Prasannadi Kashayam. Thank you. Thank you, ma'am. You are muted.
Ma'am, you are not audible. Ma'am, please unmute yourself.
I want to ask you, what is the difference in UPLC study of steroids and alkaloids?
Sir, UPLC study not UPLC study, using UPLC, I identify the compounds present in the kashayam.
So what are the characteristic difference between the two that you observe from the studies?
From kashayam, we have most of the natural compounds, alkaloids, phenolics,
Oh,
and flavonoids.
okay.
Steroids have some side effects
Mhm.
for long-term usage.
Okay. Thank you.
Thank you, ma'am.
Now the next candidate is Soumya.
Yes, ma'am. 1 minute. Ma'am.
Ma'am,
Girija, you stop sharing. Yeah.
Sunita ma'am. Hello.
Yes.
Ma'am, Prachi Singhal has requested for her presentation earlier because she
Okay.
had to leave. She has bus at 5.
Okay. Okay. We can ask them.
Prachi,
Yes, she can.
Prachi Singhal.
Yes, sir. I'm sharing my
Yes.
screen.
Yes,
Okay.
please start.
I think Girija, yeah. Girija has to stop sharing. Then only her will be visible. Yes, it's visible.
Uh,
You can start, Prachi.
Good afternoon, everyone. I'm Prachi Singhal from Meerut College, and my topic for presentation is removal of acid orange dye using banana peels as an adsorbent. So the contents are introduction and the materials and methods we have used for the process and results and their discussion, that is effect of contact time, bioadsorbent dose, and the influence of bioadsorbent dose, effect of pH, effect of temperature, and then conclusion. So firstly, going through the introduction, textile dyes and printing industries are renowned for releasing colored effluents containing toxic dyes into the water bodies, and there are so many methods available for the color removal from wastewater. But those techniques have many limitations like sludge production, residue production, excess usage of chemicals, accumulation of sludge, and less effective color reduction. So we have from the literature survey we have find out that the adsorption technique is found to be the best efficient technique for the removal of the pollutants. It is based on the transfer of pollutants from solution to the solid phase. The superiority of this method over other methods can be given in the terms of cost, design, ease of operation, non-toxicity of adsorbents comparison to the other methods. And the dye I have used is acid orange 7. It is a commonly used azo dye, but and it is problematic to the ecosystem. The study begins with the comprehensive investigation of adsorption efficiency of banana peels. That is, I have used banana peels as the adsorbent, and I conduct batch adsorption experiments for the same to determine the impact of different factors like what was the adsorption process, what was the initial concentration of dye, and what is the effect of contact time, what was the dose of adsorbent, and the effect of pH, in which pH was systematically changed and the changes were recorded as well. Now, the aim of the study is to investigate the efficiency of banana peels as an adsorbent for the removal of acid orange 7, and to identify the optimal conditions like pH, temperature, contact time, dose for maximum dye removal efficiency. So also we are going to use banana peels. Those are eco-friendly, and they have the potential to reduce the environmental pollution. Significance of my study is to contribute to sustainable and eco-friendly development of development of some system that may be utilized to absorb the harmful and hazardous dyes from the wastewater. Next, acid orange 7 dye. Acid orange 7 dye, it is an azo dye, an ionic azo dye. The IUPAC name is sodium 4 2 hydroxyl 1 naphthyl azobenzene sulfonate, and the structure is shown at the right. Its molecular mass is 350.3, molecular formula C16H11 N2NaSO4. Orange colored powder होता है, soluble in water, and it is used for dyeing various materials like nylon, silk, wool, etc. It is highly toxic, and its ingestion may cause eye, skin irritation, mucous membrane irritation, and upper respiratory tract irritation. Its intake may cause cancer because it is carcinogenic in nature. And the lambda max is 542 nanometer for the dye. Next, banana peels as an adsorbent. Adsorption actually is a phenomena where molecules or ions from the fluid adhere to the surface of a solid or liquid. And as it is shown in the figure, that process of adsorption, banana peels are the common organic waste we can use for adsorption. These peels possess a porous structure and functional groups on their surface that make them suitable for absorbing various contaminants from the aqueous solutions. Banana peels are often discarded as waste, making their utilization as adsorbent a sustainable and eco-friendly approach to waste management and environmental remediation. They are exceptionally low cost adsorbent since they are readily available as waste material from banana consumption. So it is an attractive alternative uh, to conventional adsorbent that are expensive in nature. So it is a cost effective product that we can make banana peel. We can use banana peel as an adsorbent. Next, methodology. Uh, the method by which an adsorbent using banana peels was prepared. So, this is the method. I have taken raw banana peels, then I have dried them, then grinded with the mortar and pestle, and the
Prachi, show your
coarse.
result. Time is going.
Okay, ma'am.
Show your result.
Now moving to the results, effect of contact time. So as we can see that the adsorption percentage is increasing as the contact time is increasing, and 90 minutes we have checked over the period 10 to 90 minutes, and the maximum removal of acid orange 7 on the bioadsorbent surface was observed to be 74.94% around. Next is the effect of adsorbent dose. So we have changed the dose of adsorbent from zero point 0.3 to 2 gram for 60 minutes, and we have taken 30 ml of dye solution. As we can observe that 1 gram adsorbent dose has given the maximum adsorption for the same. Next is the effect of pH. As we can see that with increasing pH, the adsorption is decreasing throughout, as acid orange is an anionic dye. So the percentage removal was 142% around when the pH was 2 to 3, and after 4, it is decreasing continuously. Next is the effect of temperature. Uh, Effect of temperature on banana peel powder. Studies were carried out between 30 degrees Celsius to 60 degrees Celsius. There were four temperature we have settled down. The results indicate that a raise in temperature decreases the AO7 adsorption as shown in the figure, and trend suggests that the of adsorption of dye on banana peel powder is exothermic. As we can
Now I
see that.
think you stop. Time is over. Conclude finally
Okay, ma'am.
and stop.
And the final conclusion is we can say the adsorption of acid orange on prepared adsorbent decreases with increase in temperature, and it is exothermic in nature. And we can say the study provide a green eco-friendly bioadsorbent banana peel powder which is efficient for dye removal and may be tested for remediation of various pollutants in contaminated industrial wastewaters. Okay, thank you, ma'am.
Have you calculated delta H also? for absorption?
मैम not till अभी तक तो नहीं उसको कैलकुलेट किया
Okay,
है.
okay.
Actually the work
Any
is
question?
ongoing.
Okay. Any question? Anyone wants to ask some question? Okay, Prachi, thank you so much. You
Okay,
can
ma'am.
stop
Thank you so
sharing your
much.
screen. Now the next candidate, Soumya.
Yes, ma'am. 1 minute.
Soumya, please start.
Yes, ma'am. Okay. Ma'am, is it visible?
Yes, yes, visible. You can start.
Okay. Yeah. So, ma'am, title is Apple Pomace Optimization and Eco-Friendly Perspective. So, I'm PhD scholar from Nifton. So, next slide. Yeah. So, ma'am, Apple Pomace is a huge global concern because jam, juice, cider industry, tons of waste is generated, and we need to utilize it in an efficient manner by nutraceutical production, by making of cosmetic products, by biochemical production like lactic acid, biosuccinic acid. So this is a review. So no such data is there, only literature is there. So all these generation will lead to ultimately better utilization of waste. A circular bioeconomy needs to be achieved in this trend of sustainability. So this is a graphical abstract. So concept of biorefinery, which our country can opt to, because next step is biorefinery development at an industrial scale. How rich apple pomace is? Storehouse of valuable compounds, proteins, fats, carbohydrates, minerals, complex carbohydrates are there, minerals are there. So this is the composition, proximate composition. So actually my main title, my research project title is based on anti-diabetic potential. And I am basically developing a snack bar in my research study. So apple pomace I have taken as one of the ingredients because apple pomace is rich in florizin, it is rich in pectin, and my MTech thesis was on pectin. So I know the relevance of pectin in the pharmaceutical market, in the development of products, the health benefits it can offer. अभी तो it is being used only as a gelling agent. बट आप फ्लोरेजिन और पेक्टिन को अगर डायबिटीज के पॉइंट ऑफ व्यू से भी देखें तो बहुत ज्यादा पोटेंशियल है. और फ्लोरोजिन में तो SGLT2 ट्रांसपोर्टर्स और इन मॉलिक्यूल्स के पूरे ग्लूकोज एब्जॉर्प्शन मैकेनिज्म को चेंज करने की कैपेबिलिटी है. तो वी मस्ट एक्सप्लॉइट दिस, यूज दिस. DPPH एंड फ्रैप वैल्यूज एंटीऑक्सीडेंट मेजर करने के तरीके. So introduction, yeah, phenolic compounds. These are all the phenolic compounds. References are listed. Yeah, a circular bioeconomy G20 summit is always it's directing towards it, and we must go with the flow. So food and feed utilization, yes. Novel approaches में fortified products का बहुत literature है. Apple pomace fortified cookies, biscuits, noodles, LCA studies. I did an internship through Khurana scholarship at Purdue University. 3 months I was there. एंड तो एलसीए का लिटरेचर पढ़ने का बहुत मौका मिला. तो बायो रिफाइनरी डेवलपमेंट के बाद सस्टेनेबिलिटी चेक करने के लिए, एनालाइज करने के लिए एलसीए का एक बहुत बाहर के देश यूएस यूज कर रहा है उस चीज को. तो वी शुड एक्चुअली ट्राई एंड एक बार बायो रिफाइनरी डेवलप हो जाए तो इन्वायरमेंटल सस्टेनेबिलिटी चेक करने का अच्छा तरीका है एलसीए. So valorization routes, biochemicals, biomolecules, functional foods, cosmetic products, bioplastics. Ma'am, this is a figure which I made myself. कहीं से ऐसा adapted नहीं है. So ये AP polyphenols का value दिखाता है कि how we can use it और क्या क्या combat कर सकता है वो skin damages में, dark spots, collagen damage, ये सब remove हो सकता है. ये मैम सस्टेनेबल एपी बायो रिफाइनरी का सर्कुलर बायो इकोनॉमी कंसेप्ट पे एक फिगर है. ये भी मैंने ही बनाया है, ये कहीं से कॉपीड नहीं है. तो इट हैज पोटेंशियल फॉर पब्लिकेशन. तो इसमें मैम एनहांस्ड बायो रिफाइनरी सिस्टम्स, प्रोस्पेक्टिव अपग्रेडेशन, जैसे मैंने कहा पेक्टिन, लिग्निन, पॉलीफिनॉल, डाइटरी फाइबर्स. पॉलीफिनॉल का मैम सबको पता है, डाइटरी फाइबर भी रफेज प्रोवाइड करता है तो वो भी सबको पता है. लेकिन पेक्टिन, लिग्निन ये सब भी आप यूज कर सकते हैं एप्पल पोमेस से हार्नेस करके. एक्सपेरिमेंटेशन एंड एनालिसिस इनकॉर्पोरेटिंग नॉवल मेथड्स, रिसर्च इनपुट एलसीए के फॉर्म में. एलसीए से पहले रिसर्च इनपुट, फिर एलसीए से एनालिसिस. देन ये मैंने भी कुछ नया देखा, थोड़ा कैलकुलेशन का पार्ट है कि एक बार सस्टेनेबल एपी बायो रिफाइनरी डेवलप हो जाती है तो उसके कैरेक्टरिस्टिक्स होते हैं 4: प्लेटफॉर्म्स, फीड स्टॉक्स, प्रोडक्ट्स, प्रोसेसेस. तो इन सबको
So, do conclusion. Time is over.
यस मैम. तो इससे
Conclude.
इसमें कुछ टर्म्स हैं, टीआरएल है, सीसीआई इंडेक्स है. ये सब मैम कैलकुलेट करके सस्टेनेबिलिटी को कैलकुलेट किया जा सकता है. इसमें मैम थोड़ी सी कैलकुलेशन है. फिर उसके बाद एलसीए, यस ओके, एलसीए देन yes ma'am, I am done. Thank you ma'am. Okay, I'll just go to the
Have you
conclusion.
published this book?
नहीं मैम, एक्चुअली आई एम एमिंग थ्रू योर कॉन्फ्रेंस कि ये पब्लिश होने लायक हो. और इसमें मटेरियल तो मैंने ऑलमोस्ट डाला है, लेकिन पेपर में अच्छे से सारा और भी डिस्क्राइब किया हुआ है, फिगर्स भी हैं. सो बस ये कंक्लूजन है. फिर रेफरेंसेस हैं,
So
रेफरेंसेस इसमें
is it
कम है
your report?
और
Is
पेपर
it
में
your
और भी हैं.
report
सो
or your PhD work?
मैम
Is
ये
it your
रिव्यू
PhD work?
आर्टिकल है जो मैंने पढ़ के लिखा है. हाँ मैम, वो स्नैक बार बनाई है, स्नैक बार में एप्पल पोमेस यूज किया है. और निफ्टेम आने से पहले एप्पल पोमेस और ये
Okay,
एमटेक
okay.
थीसिस इसी पे थी.
Anyone want to
तो
ask
फिर
any
उसी चीज को
question?
यहाँ पे यूज करके.
Okay, thank you, Soumya. You can leave the screen now.
थैंक यू मैम, थैंक यू सो
Next
मच.
candidate, Anita.
Yes, ma'am.
Yes. Be particular about time because we are running short of time. Okay?
Yes, ma'am.
Yeah. You have to be fast. Try to do the main part
My
within
screen,
5 minutes.
my screen visible?
No. I think first of all, Soumya has to leave. Soumya, stop sharing.
Hello.
Yeah, now Anita,
Yes, ma'am.
you can start.
Ma'am, my screen is visible?
Yes, yes, it's visible now.
Oh, okay. Ma'am. One second.
Anita, please make it full screen.
Yes, ma'am. I am Anita Dubey, research scholar from Department of Environmental Study. My review article entitled Wastewater Remediation Using Plant-Derived Zero-Valent Iron Nanoparticles. In this presentation, we will discuss the green route of synthesis, iron nanoparticles using plant leaf and their impact on the removal of wastewater pollutant. Now we discuss some basic terminology. What is nanoparticles? Cluster of atom and or molecules having size 1 to 100 nanometers are called nanoparticles. When discussing plant-mediated process, it is important for us to understand what green chemistry is. Green chemistry is a branch of chemistry that focus on designing and developing process and products that are environmental friendly, cost effective, sustainable, and reduce or eliminate that use and generation of hazardous substance. Green nanoparticles has superior property than bulk substance. These properties are mechanical strength, thermal stability, catalytic activity, electrical conductivity, and magnetic property. That is why nanoparticles are used for the removal of heavy metals and dye and etc. Leaf extract has been demonstrated to provide some of the best capping and stabilizing property for the synthesis of nanoparticles, which have shown great efficiency is waste. And why leaf substance are used? Because of eco-friendly nature, cheaper process, and varied shape and size, photochemical, antioxidant, and abundant production. Now, moving on, objectives, serving leaf litter scenario in Delhi and estimated their potential toward nanoparticles synthesis, identification, and characterization of synthesized iron nanoparticles, and qualifying nanoparticle efficiency in degraded dye and toxic metals. Hello. In this study, first we will conduct a survey of the area, then we will proceed with the synthesis of nanoparticles. After that, we perform characterization to confirm the formation of nanoparticle. Finally, we treat the waste and test its efficiency. Now, when we conducted survey in Delhi, Delhi has arid and semi-arid zone have a thorny scrub vegetation, and the campus of Delhi, having different colleges, is blessed with rich vegetation and many departments poses a compulsory green belt. The most abundant tree species in Delhi colleges is Syngenium pumini, 28%, and Polyilthia longifolia accounting for 24%, while the least abundant species in Ficus religiosa, that is people, representing 12%. And now processing of leaf to make iron nanoparticle, there are several methodology, and one of these explaining here. Leaf litter, we are collecting separate, weight and washed thoroughly and dried in an oven to remove all moisture of leaf. Dried leaf were then crushed and powdered to make the extract. Then proceed aqueous leaf extract was added into ferrous chloride solution, and after a while the color of the solution changed from pale yellow to intense black, and which indicate the formation of iron nanoparticle. And this solution dried to hot plate convert into black solid precipitation and crushed and dispersed into fine particles. And characterization of iron nanoparticles, various characterization techniques such as SEM, TEM, FTIR, XRD are used to understand and access the property of iron nanoparticles. TEM and SEM, this technique provide detailed information about the size, shape, morphology of iron nanoparticles, and XRD is used to determine the crystal structure of iron nanoparticle. It reveals information about their crystallinity, phase composition, and lattice parameter. And FTIR help identify the chemical composition and functional group present on the surface of iron nanoparticles. And zeta potential measurement access the surface charge of iron nanoparticle. This parameter is vital for understand for stability, dispersibility in solution, and the interaction with with other materials. And double AS and the EDX chemical composition reveals the atomic element which nanoparticle is composed. And double AS and ICPMS, this is done to measure the degree to which material dissolved for a nanoparticle. And next, we have summarized all the research articles related to iron nanoparticles synthesized using leaf extract, focusing on their application in the removal of the heavy metals and dye, compiled there in this table. Major finding of this review, according to this table, numerous paper has been published on chromium removal, while the least number of paper has been published on cobalt removal. The most common nanoparticle size study fall within the range of 15 to 16 nanometer, and significant percentage of the study achieved 70 to 100% removal rate within 1 to 6 hours. Notably, 100% removal was observed for methylene, orange, and chromium. Additionally, green tea and eucalyptus leaf were the most commonly used material in this study. In this study, salient features are iron and particle have great efficiency, is remove dye and toxic metal. Plant-derived zero-valent iron nanoparticle green synthesis is now evidenced to be relatively safer, sustainable, and the least reported or no reported toxicity. The size of nanoparticle play a significant role in degrading dye and toxic metal sample. The size range of iron nanoparticle is most study is 10 to 60 nanometer. Leaf extract has been demonstrated to provide some of the best capping and stabilizing property for the synthesis of nanoparticle, which has shown great efficiency in waste treatment process. During our survey, we found that many tree species such as Pongamia pinnata, Gilia africana, Syngenium cumini, etc. are abundant in...
Your time is over now. We can stop now.
Okay, ma'am.
Why are you doing this type of studies? Are you doing some experimental work also?
No, ma'am. अभी मैम स्टार्ट किया एक्सपेरिमेंटल वर्क विद डाई के साथ और लीफ वेस्ट के साथ बेस्ड ऑन लिटरेचर रिव्यू. In the most of the research articles, ma'am, synthesis has been carried out using fresh leaf, but my study utilized dried leaf waste for synthesis.
Oh.
Oh.
Thank you.
Okay, thank you.
You can stop sharing now.
Okay.
Next, Anuja. Anuja is here.
Yes, ma'am, I'm here.
Okay.
Ma'am, is it visible?
No. We have just 5 minutes, Anuja. Within 5 minutes, try to conclude.
Is it visible, ma'am?
Yes, visible.
Okay. Okay, good afternoon, everyone. My topic is physico-chemical analysis of groundwater in district Telangana. So introduction of my topic is water is an inorganic, translucent, tasteless, unscented and colorless fluid, which is the chief element of Earth hydrosphere. It is crucial for all forms of life, even though it provides no calories or organic nutrients. Quality of water plays important role in crop productivity, not just for crops, but for human beings also. Quality of water plays an important role. In our research, we will study about the physico-chemical properties of water. Water travels in the body along with nutrients and minerals. Water keeps our body cool as part of our body's regulating system. Water quality refers to the chemical and biological characteristics of water based on the standards of its usage. We will be collecting water samples from different blocks of Secunderabad district and will be doing water analysis there. So objectives are to figure out the water chloride level of Secunderabad district of Telangana, to notice chloride level of various drinking water sources, to examine the various physico-chemical water quality parameters that are pH, electrical conductance, TDS, total hardness, calcium, magnesium, total alkalinity and carbonate, sodium, potassium, sulfate and nitrate. To forward water quality awareness in school along with photo documentation, to suggest the remedial measures for the removal of chloride. Methodology is as follows. First of all, we will be doing sampling. The analysis will be carried out by collecting groundwater samples and by analyzing them under laboratory conditions. Second is parameters. The various physico-chemical parameters would be pH, electrical conductance, TDS, total hardness, sulfate, nitrate, phosphate, calcium, and will be analyzed by following APHA 1995. Chloride in water samples will be analyzed. Comparison of excretion of chloride from urine of respondents from minimum and maximum chloride in groundwater from study area. Awareness. At block level, amongst school children, guidance should be provided to ensure that school children get adequate access to water. Spectrophotometer. A UV spectrophotometer measures the intensity of light passing through a sample solution in a cuvette and compares it to the intensity of light passes through the sample. Then flame photometer is an analytical instrument used in clinical laboratories for determining of sodium, potassium, lithium, and calcium ions in body fluids. Gravimetric analysis. This method determines the chloride ion concentration of a solution by gravimetric analysis. Result and
Explain your result. Huh?
yeah, ma'am. Result and discussion. A pH of 7 is neutral. We all know that pH 7 is neutral of water. A pH value below 7 is acid and above is alkaline. The pH value between 6.5 and 8.5 is considered normal. Most of the samples that were collected and tested were safe for drinking purpose. Few samples were acidic with a lot of impurities in them, which were not safe for drinking purpose. Different samples had different values of pH, hardness, chloride concentration, magnesium, and electrical conductance. On the basis of different values, we found out that some of the samples were fitting into the category of safe drinking water, whereas a few samples were not safe for drinking purpose. Thank you.
Any question? Okay, thank you. Now the next
Thank you, ma'am.
candidate is Ranjita.
Yes, ma'am.
Yes, we can start.
Good afternoon, everyone. Myself, Ranjita Singh, a PhD research scholar from Department of Chemistry. Today, I am going to present my work on the topic Synthesis and Application of Nanocurcumin towards Methylene Blue Degradation in Aqueous Medium. Contents: Introduction, Materials and Method, Results and Discussion, Conclusion and Future Scope, References. So what are nanoparticles and what is nanocurcumin? Nanoparticles are particles whose size ranges between 1 to 100 nanometer. Depending on the overall shape, these materials can be 0D, 1D, 2D, or 3D. The interest in synthesis of nanomaterials has grown because of their distinct optical, magnetic, electronic, mechanical, and chemical properties compared to those of the bulk materials. What is nanocurcumin? Nanocurcumin are modified form of curcumin and it has small particle size. Photocatalyst and its effect in dye degradation. What are photocatalysts and how they impact on dye degradation? Photocatalysts are materials which can easily alter solar energy for use in oxidation and reduction activities. They've been a topic of great interest during the last few years because of their ability to remove toxic and hazardous substances from the environment and its dye degradation. And the highlight features of photocatalyst are appropriate band gap, good photostability, high carrier mobility, correct bandage position, high surface area, efficient light absorption. Objective, the main objective is the synthesis of nanocurcumin by ultrasonication method, that is top-down method within 100 nanometer range. So top-down approach basically involves the formation of nano-sized structures by breaking down the bulk material. Top-down approaches are inherently simpler and depend either on removal or division of bulk material. And the second is synthesis of photocatalyst and further use it in the degradation of dyes. So photocatalysts are materials which can easily alter solar energy for use in oxidation and reduction activities. So the plan of work is synthesis of nanocurcumin by top-down method and combine it with zinc oxide and nano zinc oxide to prepare photocatalysts which are characterized and used to degrade methylene blue dye. Materials and methods. So these were the instruments and reagents used during our work, namely curcumin, magnetic stirrer, which is used for stirring purpose, ultrasonic cleaner, which is used for sonication purpose, ultraviolet fluorescence analysis, cabinet, which is used for photodegradation of dye, and these are the reagents, namely curcumin, which is in keto form and one is in enol form, methylene blue, twin 20, which is also known as polysorbate 20. Methodology, diagrammatic representation of preparation of nanocurcumin by ultrasonication method. So the curcumin nanoparticles were prepared by the technique described by Poreza with few modification here. So the procedure performed was as follows. The organic phase was prepared by dissolving 20 milligram of curcumin in 25 ml of DCM, that is dichloromethane. And now for the preparation of aqueous phase or liquid phase, 10 ml of twin 20 and 5% volume by volume was added to 95 ml of double distilled water to prepare 5% twin 20 solution. And 10 ml 5% twin 20 was added to 90 ml boiling double distilled water to prepare aqueous phase. Then 2 ml of organic phase was added dropwise to the aqueous phase, about 10 drops per minute under ultrasonic conditions with an ultrasonic power of 50 watt and a frequency of 40 kilohertz with temperature 80 degrees Celsius with 40 minutes for 40 minutes. So the whole solution was stirred at a magnetic stirrer with high rotation for 40 minutes until a yellow colored solution was obtained. So we used a rotatory evaporator to evaporate the dichloromethane, and pH of the solution was measured and then characterization was done. And this is the diagrammatic representation of preparation of nanocurcumin zinc oxide photocatalyst and nanocurcumin nano zinc oxide photocatalyst. So here 10 ml of prepared nanocurcumin solution and 0.5 gram of zinc oxide was taken in a beaker containing 10 ml of water and 10 ml of ethanol. Further, the mixture was sonicated for 1 hour, followed by magnetic stirring for 1 hour. Then the resulting precipitate was filtered and washed with water several times. And the product was then heated in an oven at 100 degrees Celsius for 3 hours, and nanocurcumin zinc oxide photocatalyst using curcumin was prepared. This is the UV visible. This is the result and discussion part. UV visible study. So in this figure, curve A represent the visible UV visible spectrum of raw curcumin, and B represent the same for the prepared curcumin solution. So from this figure, it is observed that a absorption peak is noticed at wavelength 420 nanometer in case of nanocurcumin, which is depicted as B, whereas slight peak is observed in case of raw curcumin. So from this study, it can be said that nanoparticles of curcumin is formed. This is the XRD analysis of nanocurcumin zinc oxide and nanocurcumin nano zinc oxide photocatalyst. So from the figure A, XRD analysis, it was found that zinc oxide nanoparticles were formed, and this was proved by the sharp peaks which symbolize that crystallite structure zinc oxide nanoparticles.
Try to conclude. Time is over, Ranjita.
Yes, ma'am. This is the FTIR analysis of curcumin zinc oxide photocatalyst. And these are the dye degradation of methylene blue. This is the degradation percentage of methylene blue of curcumin at different time. And these are the degradation percentage here. So from the data mentioned above, it was found that methylene blue was degraded by 70.77% in 1 hour 30 minutes by nanocurcumin zinc oxide photocatalyst under UV light irradiation, and 63.69% was degraded in 1 hour and 30 minutes under sunlight in the effect for the same photocatalyst. But methylene blue was degraded by 86.96% in 1 hour and 30 minutes under sunlight in the effect of nanocurcumin, nano zinc oxide, nanophoto-catalyst. So conclusion and future scope. So here we have synthesized curcumin nanoparticles within 100 nanometer range by top-down method, which shows good water stability and high stabilities. The prepared nanocurcumin was combined with zinc oxide and nano-zinc oxide to prepare photocatalyst, which proved to be efficient to a great extent in degrading methylene blue dye. So out of the two photocatalysts, the nanocurcumin nano-zinc oxide photocatalyst degraded methylene blue up to an extent of 86.96%, which may be due to the increase in surface area after the formation of zinc oxide photocatalyst. And future scope is in the application of prepared nanoparticles as pH sensor for different water sources, and also it can be used in the application of synthesized photocatalyst for the degradation of other dyes as well. These are the references. Thank you.
Okay. Any questions?
Why did you choose curcumin? Because it has some caste.
Yes,
Why did
because
you?
curcumin has fluorescence properties, and it absorbs in visible regions, so it becomes easier for analysis.
But it will cast more, na, for dye degradation.
Yes, sir.
Okay, that's it.
Thank you, sir.
Okay, thank you, Ranjita. You can stop sharing now.
Okay, ma'am. Thank you, ma'am.
Next is Vijeta.
Ma'am, am I visible?
Yes.
Good afternoon to all of you. My name is Vijeta, research scholar at BMU University, Rohtak. My topic is chemical and pathogenic analysis of drinking water in the district Rohtak, Haryana. Introduction consists of water is the main constituent of Earth hydrosphere and the fluids of all living organisms. It is important for all forms of life on Earth. In spite of availability of such a huge quantity of water on Earth, the water which can be considered as fresh and can be utilized directly for drinking purpose is very limited. It is an excellent solvent that dissolves most minerals that come in contact with it. Therefore, in nature, water always contains chemical and biological impurities that is suspended and dissolve in organic and organic compounds and microorganisms. These compounds may come from natural sources and leaching of water deposits. Our water resources are being polluted at a very fast rate. The WHO reports that approximately 36% of urban and 65% of rural Indian were without access to safe drinking water. Due to increased industrialization, human population, use of fertilizers in agriculture, and man-made activities, it is highly contaminated, which results in undesired changes in physical, chemical, and biological characteristics of water. Here are the parameters that are to be analyzed in water analysis. There are three types of parameters: physical, chemical, and biological. Physical parameters consist of color, odor, temperature. Chemical consist of metals, sulfate, carbonates, bicarbonates, sodium, potassium, calcium, magnesium, and fluoride. Heavy metals like lead, mercury in water causes serious problems like damage to brain and kidney, bloody diarrhea, abdominal pain, even in trace amounts. Another constituent, nitrate, if present in water, can cause a serious problem named methemoglobinemia. Symptoms like rapid rate, heart rate, headache, nausea may appear. Biological parameter consist of bacteria, algae, and protozoans. In terms of direct effect on human health, biological parameters are more important than physical ones. Human beings and other animals discharge large number of intestinal bacteria from stool and urine. The bacteria therefore appear in drinking water when the water source is contaminated with stool. The illness caused by microorganisms like viruses and bacteria that are ingested through contaminated water is called waterborne diseases. Some
Show your
waterborne
result. Vijeta, show your result.
Among the analyzed samples, apart from physical and chemical parameters, the pathogenic ones were also found crossing the permissible limit and therefore failing the quality check. According to WHO, zero count of E. coli per 100 milliliter of water is considered safe for drinking. E. coli is a strong indicator of sewage or animal waste contamination. Some waterborne pathogenic microorganisms spread by water can cause severe life-threatening diseases like typhoid, cholera, and hepatitis. Among all the waterborne diseases, the major and utmost concern are diarrheal diseases causing high mortality in a large number of developing countries. Diarrhea is commonly a symptom of intestinal tract infection caused by a variety of bacteria. The most causative microbe for diarrhea, which include E. coli. Our dependence on fresh water resources has accelerated in last centuries due to fast growth in world population. To lead a healthy and safe life, water quality assessment is the need of hour.
Any question? So from which place you have taken your sample?
Ma'am, in Rohtak district, various blocks.
So which water is, according to you, which water is more free from impurity?
Ma'am, free from impurity.
Yeah. Which region water is more
Ma'am,
free
among
from
rural and urban, if we compare urban and rural water, then urban.
your result? I'm asking about your result. What is your observation?
Ma'am, urban water is more.
Okay. Thank you. Any other question? Anyone want to ask anything? Okay, Jan, on your screen. Next is Ekta.
Ma'am, I think Ekta is not present.
Ekta is not here. Then next is
Ankita.
Ankita. Ankita.
Yes,
She is
ma'am, I am here. I'll start.
yes. Yeah.
Ma'am, is my screen visible?
Yes, yes, it's visible.
Okay, now also?
Yes.
Okay, ma'am. Good afternoon, everyone. My name is Ankita. I am a student of Environmental Studies, Delhi University, and I'll be presenting my presentation on estimation of biosol concentration and associated potential risk in the institutional environment, a case study from urban city of Delhi. It has been reported that the world's 99% population breathes air exceeding WHO guidelines. This air is contaminated by the pollutants, largely categorized as physical, chemical, and biological agents. While the physical and chemical air pollutants are studied extensively, the study on biological contaminants is still in its infancy stage in India. The biological contaminants or the biosol are the minute biological material omnipresent in air, which can be pathogenic and non-pathogenic. They can be fungal, bacterial, pollen, viruses, and byproducts of the microorganisms, including endotoxins, mycotoxins, butabiquins, etc. The sources and transmission of biosol includes the natural sources like flora, fauna, industrial, building, waste management systems, disaster and terrorism, and the transmission is majorly occurring by natural sources like winds, oceans, water, and people-to-people interaction, and the combination of all of the above. Factors affecting biosol levels includes the temperature, solar radiation, relative humidity, wind speed, precipitation, atmospheric pollutants, and the design of building and the infrastructure of the building. Now, the exposure of the bioaerosol may occur through three main mode of infection, that is through inhalation, contact, and ingestion. Now, these are some of the health effects caused by majorly occurring bioaerosols. I'm going to skip this. Now, the importance of studying bioaerosol overall is because of the following 5 reasons. They are easily carried by air currents. They are dispersed in form of inhalable particles, and they are omnipresent. And it has been reported by a study by Kumar et al. That in operation theaters, bioaerosol concentration was found. Although it was low, but there was contamination. The bioaerosol deposits in the various parts of the body through lungs and circulatory system. And a study from Jong et al. Has suggested that the bioaerosol deposits in the respiratory system and because of their minute submicronic sizes, they are able to deposit in the alveolar region also. They are proven allergen and pathogens and causes many respiratory disorders, hypersensitivities, and other diseases. And the worst part is that there are no universal guidelines regarding the bioaerosol contamination. There are guidelines set by many governmental and private agencies just for their purpose and not for the universal. Here are some of the guidelines. Now, I have chosen the site as educational institutes. The reason of the educational institutes being selected as a site is because the majority of the Indian population is in the age group of 0 to 24 years, which is the most studying population. And the college students, from a survey conducted by us only, has reported that the students spend more than 8 to 10 hours of their day in the institutional campus, be it indoors or outdoors. So the objective of my study was to determine the bioaerosol load in different microenvironments of the institutional area, education institution, using the traditional passive plate method. The methodology of my work was to select a site in Delhi University, and there were 10 sites, 7 indoors and 3 outdoor sites that were selected for this study. The air samples were collected on nutrient agar plates, conventionally through gravitational method, and the samples were incubated for 37 degrees for 24 to 48 hours until the colonies were formed, and the colonies were counted using plate count method. The on-site temperature and relative humidity was also assessed using handheld crystal body monitor, and the bioaerosol load was reported in CFU per meter square per minute. These are my on-site details of the different sites: the average footfall, the ventilation, the temperature, and the relative humidity. These are my results. So, graphically representing my results, as it can be seen that in the indoor sites, with the first 7 sites in yellow colored bars, the plant tissue culture room and the biotechnology laboratory has very less number of footfalls, that were less than 5 footfalls per hour. And there was good ventilation because there was a sterilization unit also. So the bioaerosol concentration load or the bioaerosol levels in these sites were very less as compared to the sites, indoor sites like washroom, zoology laboratory, classrooms, and the cafeteria. These sites were having higher bioaerosol load because although there was ventilation, the higher average footfall per hour was high. That led to aerosolization of the bioaerosol that were on the surface of the substances or from the ground to into the air, which was ultimately collected on the agar plates. As it can be seen in the green colored bars in the outdoor sites, the bioaerosol concentration was more or less similar, although they were different sites, because the air or the wind, they have carried the bioaerosol and it has prevented the concentration of bioaerosol in a particular area, as unlike in the indoor sites. It was also found that the temperature and bioaerosol concentration has a positive correlation, although there was a negative correlation with the relative humidity. There was a positive, strong correlation between the average footfall and bioaerosol concentration. I have already discussed the results. So to conclude my study, I can say that ventilation plays a very important role in circulating the air and therefore lowering the accretion of bioaerosol in the air, which can be seen in the indoor sites with high footfall, and the lower ventilation. On-site temperature and relative humidity also plays a very significant role in on the concentration of aerosol on the sites. Also, the activities that causes aerosolation, that can be stampede, that can be footfall, or having lunch, or having eating something in a site, that also causes the aerosolization of microbes from different sources and also increases the concentration of biosol in the air. The total biosol concentration were high in all the sites, that was exceeding the WHO limit, which is 500 coliforming units per meter square per hour. So it is the need of an hour to study the exposure level of airborne biogenic particles within indoor and outdoor environment to assess the possible health risks that the students might be exposed to in agglomeration of Delhi and the built-up environments. And as there are no guidelines set for the biological contamination, as it's already known that in the AQI we
Game
only
over. Stop
and just
now.
one more line, please.
Yeah.
Yeah. As it has been already seen that in the AQI, we only take the particulate matter and the gases pollutant in calculate the air quality index. So we are trying that if we put enough of the study, if we put enough of the data in outside, so we can also set guidelines for aerosol contamination in the air. I would like to thank my professor, Chirashri Ghosh ma'am, Dr. Rekha, my co-supervisor, my fellowship, and the live staff of Environmental Studies and Miranda House. Thank you so much, ma'am.
Thank you.
Ma'am, any
Thank you.
question?
Any question? Anyone wants to ask? No. Thank you.
Okay, thank you so much, ma'am.
We have other candidates, Anil? Divyanshu and Harsh.
Devanshu and Harshvai, yes.
Divyanshu
Yes,
is
ma'am.
here?
Yes, ma'am.
Divyanshu, you can start.
Okay. Is my screen visible, ma'am?
Yes.
Yes.
Okay.
Divyanshu, be particular about time, only 5 minutes.
Okay, ma'am. Okay, sure. Good afternoon to everyone present here. Today, I'm here to present my work on the development of curcumin as a modified cotton for potential application removal of lead from water. So the major sources of lead that is contaminating our water and our lands is basically industrial mining and agriculture. They play the most crucial part in contaminating the environment, and the effect the lead has on the body is very severe when it is accumulated for a long time, as it is evident from these pictures here that it affects almost all the organs that we have in our body. Now, what are the treatments that are available for the heavy metals uh, diseases? So basically, if you look at the chart here, one thing that is very similar in all this is one name is recurring more than others, and that name is curcumin. So curcumin has a very abundant in this picture, as it is very useful in treating the heavy metal modalities. Now, why does curcumin have this kind of abundance here? The answer is basically because it has such a chelating potential with heavy metals, be it divalent metals, trivalent metals, or trivalent metalloids. The curcumin has a binding efficiency with all of them, and it binds strongly with the heavy metals. Now, the rationale of our work is we have prepared a propylated curcumin and a cotton azide, and using the click chemistry reactions, we have combined them using the triazole ring. So first, what we have done is we have propylated the curcumin using a propylyl tosylate in the basic medium for 3 to 4 hours at RT, and we have modified the cotton with the azide, and then using the click chemistry reactions and conditions, we have been able to attach both of them using a triazole ring. So the synthesis start basically with the propylyl alcohol, because to prepare the propylated curcumin, we first have to synthesize the propylyl tosylate. So we use the starting material propylyl alcohol reacted with it tosyl chloride in a basic medium, and then we observed the product here, which is propylyl tosylate, which is a dirty yellow in color, and the confirmation of the product being formed is done by using a TLC initially, and then we performed a NMR study on this, where we can see the proton signatures matches with that of our structures. And here at 7.7 and 7.4, we get a spectrum that is very similar with the para-substituted molecules. After that, we have confirmed that the propyl tosylate has been formed. We modified the curcumin with the propyl tosylate in basic medium at room temperature for 3 to 4 hours. And then we obtained the propylated curcumin, which is the propyl handle that we'll be using in the click chemistry. Again, the confirmation of the formation of a product is again done by TLC initially, and then we again did the NMR study of this. Here also the proton structures of our spectrum matches with that of our molecules. At 2.45, we have some abnormalities because the undetected proton of the DMSO merged with our peaks, that is the HA peak. Other than that, we have a very clear picture of the proton signatures here that correlates with our structure. Then again, we confirmed it with mass. We got the M plus
Show
1
your
structure
result,
of 4.
beta. Show your result. Time is over.
This one, ma'am?
Result. Show
Okay,
me your
sorry,
result
sorry.
and conclusion.
Okay, so then we basically did the click chemistry reactions through the cycloaddition, and this is the picture of our product. Then what we did is estimated the lead in-house, and we used a 10 ppm solution of lead solution, passing it through our cotton and for 15 to 20 minutes, and we observed a decrease in concentration, and the observed concentration was 1 ppm. So using 0.5 gram of curcumin, we were able to reduce 9 ppm of the concentration of the lead. So in conclusion, what we have done is we have prepared a method for covalently linking polymer curcumin and azide using a cycloaddition alkyl azide copper catalyzed reaction. Then we confirmed the synthesis of a propyl tosylate and curcumin using NMR and mass spectra. Further studies, as this is still a ongoing process and the development is under process, so we'll be again doing some more studies like AAS and ICP-MS to further support our claims. And also we will be moving on to other heavy metals like iron, mercury, arsenic to see if it has the potential to bind with them as well. These are the references, and thank you, ma'am. Thank you, everyone.
Why only curcumin? Why you have used curcumin here? Why not any other chemical?
Basically two reasons for using curcumin. One is that it was very abundant in the literature review that we did with the binding with heavy metals. And the second one is if it is leaches out from the cotton and goes into our system, it has very less effect or very less harmful effect. I don't think we have any reported harmful effect that curcumin has on our body. So even if it leaches out, it won't be harmful for us, which is not very similar with other chelating agents. Thank you, ma'am.
Thank you. The last candidate.
Yes, ma'am. Yes, ma'am.
Within 5 minutes, you have to complete.
Okay, ma'am. I'll try. Is it visible, ma'am?
Yes, it's visible.
Okay, thank you, ma'am. Okay, good evening, everyone. So my topic is study of nanostarch based on biodegradable films, a review. So we all know plastic have very harm effect on our environment. So I'm skipping these slides because we have shortage time. Yes. So due to bad impact on environment of plastic, biodegradable plastic get introduced to world. The development of biodegradable plastic, which are environment-friendly products, are very obtained. Bioplastic films are more sustainable product and improving the carbon footprints. They consume less energy, easily manageable, reduced waste, lower petroleum consumptions, reduction of toxicity to the environment, and when they decompose, they do not release any harmful products.
So the types of bioplastic: starch-based bioplastic, cellulose-based bioplastic, protein-based bioplastic, and some aliphatic polyesters. So basically, the most suitable group of biopolymers employed in the manufacturing of biodegradable films are polysaccharides and proteins. Polysaccharides like, as we discussed before, starch, cellulose, chitin, chitosan, and alliginic acid of natural origins are gelatins. These are the structures of starch, cellulose, chitosan, and chitin. So starch. So my work is basically on the starch. Starch, the main component of starch are amylose and amylopectin. Amylose is a linear chain linked with alpha-1 glucosidic bond, whereas amylopectin is a highly branched macromolecule consisting of short amylose moieties linked via alpha-1,6 glucosidic bond. So these polymer create a semi-crystalline structure that result in starch granule of different size, including 2 to 100 micrometers. So many work has done on starch. So I basically tried to synthesize nanostarch. So firstly, what are nanostarch? Nanostarch has a size basically 1 to 100 nanometer, has become the focus of numerous study and has received increasing attention from public because of its unique surface area and reactivity of compared to native starch granules. Its property, their superior mechanical properties, barrier properties, thermal properties at low levels, their recyclability, transparency, and low weight. Method of preparation. There are various methods of preparation of nanostarch. So acid hydrolysis, enzymatic physical treatment, combination of hydrolysis and ultrasonication, nanoprecipitation, emulsion cross-linking, enzymatic hydrolysis, and precrystallization. So method of modification of nanostarch. Graft to strategy, grafting from strategy, oxidization, and cross-linked. Why do we modify starch? What is need to modify that? By esterification, preparing hydrophobic ends, by condensing and esterification reaction enhancing the hydrophobicity, by oxidation reaction improving and dispersibility of nanostarch, by cross-linking and esterification enhancing the hydrophobicity and providing the pH responsiveness. By cross-linking and esterification reaction improve the dimensional stability. How we characterize the nanostarch particles? By UV visible spectroscopy, TEM, transmission electron microscopy, and dynamic light scattering. So what are the characteristics of nanostarch-based composite films? Their thickness, their moisture content, vapor, water vapor transmission rate, water solubility, burst strength, and biodegradability. So applications, we can use nanostarch as fillers, emulsion stabilizer, delivery vehicles, edible films, adsorbents, binders, thermoresponsive materials, templates, biosensors, and biomakers. Problems and challenges of nanostarch. Some of the limitations are their poor mechanical properties, low crystallinity, and increased brittleness over time. So because of retrogradation, and low water stability that are not suitable for certain applications. So my conclusion is the use of nanostarch is in the starch biocomposite films provide a new opportunity to create innovative biodegradable packaging and enhance different physicochemical properties. Future scope, the future study on a better understanding of nanostarch exposure level is required to solve non-biodegradable film issue. Research on nanostarch material should also be conducted to close the regulatory, financial, and technology gaps between conventional and sustainable packaging used in food industry. Thank you, ma'am. That's all from my side.
Okay, thank you.
हेलो मैम।
जी
हाँ मैम, ये जो आइसोथर्म होता है, एक क्वेश्चन पूछना था। उसमें मैं कुछ कर रहा था एक एक्सपेरिमेंट, तो उसमें 96
कौन
प्लस
बोल रहा है?
आनंद बोल।
हाँ जी,
आनंद।
okay. You want to ask candidate?
No, sir. No, ma'am, to you.
Acha.
I've seen your paper, no, ciprofloxacin ka removal. So, so in our lab also, like someone has done work with ciprofloxacin removal using biochar.
Ji.
So, ma'am,
We can discuss, Anand.
yeah,
Huh? You can take my number. We can discuss, and we have to complete this session now.
okay. So
Okay.
how do I get the number, ma'am?
I will
Website.
provide. I will provide.
Anil will provide, because this session has to conclude now. Okay.
Okay.
You can talk.
Okay. On the behalf of organizing committee ICHE 2023, I would like to thank Sunita ma'am for sparing her
Thank you, Anil.
valuable time for us, and I
Yeah.
thank all
Thank
the
you so much.
participants for participating in this conference. Thank you so much.
Thank you. I'll send the sheet, evaluation sheet. Okay. Thank you.
Okay.
Thank you, all students.
Thank you, ma'am.
much.